For many years, I ran the plant ecophysiology group at Monash University. This is my group in 2023. It's been dwindling since then, and since I, officially retired, in the sense of not getting paid. But I've also known by many people for the work I've done in teaching and educational technology, and been a bit of a pioneer in that, developing educational technology and writing programs for teaching. I was going to say online learning, but it was actually before there was the internet. So actually it was done by computer-aided learning, we called it. So there's some people in the world who that's how they know me. Other people in the world know me as a science communicator. And that's because when I first went to Monash, I actually my job was called lecturer in science communication. I'll come to that in a minute. It's a bit weird, but I actually taught science communication for about 18 years. But what I'm going to really focus on today is my nickname of the Cyanide Lady. And I borrowed the title for my talk from an article was written by Clarissa Collins and together with me called The Poison Plate of Climate Change, which among other things won the Crawford Fund Journalism Award. In fact, the first year they had that award. That's you can look them up. So that was that. Now, here's me as a little girl. And I started with this because we've just had the International Day of the Girl Child. And I think it's worth remembering that every time you see a little girl, she has a lot of potential. But every time you see a female scientist, they started out as a little girl. So to have that in your mind, I think is helpful. Now, when they had a thing on Twitter a few years ago where people put up, this is me, this little girl is me. And I chose this picture for a couple of reasons. One is because it was very girly, which I'm not always very girly, but this was a very girly dress with a frilly dress. And I'm in my garden at home with the flowers, which I always loved. And it's one of the reasons why I became a plant scientist in the first place. I also like it because I'm trying to stand like my older sisters and I never got the hang of it, of how to stand elegantly. Now, I grew up in Melbourne, and I was fortunate to have parents who were highly educated and valued education. They were both general practitioners in Melbourne, medical people. In fact, when I was this age, we still had the medical practice in the house. Not that I can remember it. And so I had this great opportunity to be surrounded by the discussion of medical emergencies and very interesting things that probably 5-year-olds shouldn't hear about at dinner every night. They were very fascinated in science. And my older sister, who's 8 years older, she had this book called Dynamic Biology as her textbook. And this was very exciting because it was in colour. Now, little beknown to the rest of my family, I used to lie in her bedroom and read this book. I mean, I must have been like 8. And I was just fascinated by all these diagrams of these animals and their positions of the organs. I even sort of got the hang of how they're related, and in my mind was sort of approaching evolutionary relationships, even though I hadn't really learned any of that yet. And I also had a few pages on plants, and she got rid of this book, but I found it about 5 or 6 years ago in a second-hand shop, and I bought it and I remembered every picture in it. So I think the value of having access to material and being in an environment that values education is incredibly important. But how did I get from being a little girl to this? And why did I do science and not medicine? Well, really, as Davo pointed out, the rest of my family are doctors, including my brothers and uncles. And if any of you are from Sydney, there's the David Adams Prize for Physiology that's named in honour of my uncle. So there's a lot of doctors on both sides of my family. So 2 things. One is, at heart, I'm very rebellious. Everybody in my family that could do science did medicine. That was the first thing. I think the second thing is, in fact, that my father died when I was in high school, and I thought I didn't really want to do what he'd been doing. But also, I just found science fascinating. I just loved being able to get to the underlying principles of what was going on. And we had a geneticist at school. Oh, actually, the teacher invited a friend who was a geneticist who came in. And I was absolutely fascinated. And I thought, yeah, I really want to do some of this. Now, clearly, there's a lot of work involved in this. And why plants? Basically, when I got to uni, the people in botany were just really just much more friendly than the people in any other department. So that would probably just sum it up. And I think all of us who in botany, who we're actually incidentally all getting together next week, would agree with that. So mostly when you study, you know, you go to school, you might do an honours degree, you might do a master's, then you do a PhD, do a postdoc, then you have some life, then maybe you go and do a fellowship, and then you get a teaching and research academic position. So that's the general path. But for me, it was a bit different. I did a master's. I could have converted that to a PhD. In fact, I got 4 papers from my master's, but I was just heartily sick of being a student. So then I went and worked in agriculture, and then I went and had some life. And it was very common in the 80s to stop work when you had a baby, and that's what I did. I nearly went mad after about 6 weeks. So then I did a lot of tutoring and so on while that went on. But it was really just sort of, I call that period life. Then I was an educator. Then I did my PhD. And then I worked in science communication, where I met various science communicators, including the one sitting in the front row here. And then I became a teaching and research academic. So a very different path. And I don't regret that, but it is different. And I also just say we've had a lot of fun on the way, and I do like to kind of ham it up a bit, as on the left in Malaysia, visiting the Malaysian campus, in the lab with my grandchildren. And actually, when I had all the students had to shake up their tubes in the lab once, I did actually put on Shake It Off and dance to it, but that's another story. Today, we're going to talk about something more serious. We're going to talk about carbon dioxide in the atmosphere and how it affects plants. Now, we know carbon dioxide in the atmosphere is rising, and it's overwhelmingly from fossil fuels. This is the Keeling curve, which is measured at Mauna Loa Observatory in Hawaii. And you can see it goes up and down every year. It goes down when all the leaves are on the trees in the northern hemisphere that draw down the CO2, and then it goes up in winter when all those deciduous trees lose their leaves. So we actually can see the influence of photosynthesis and the whole planet kind of, if you like, breathing. If you think it's going up and up and up, this is the record for the last 2,000 years. And where we are 1960 to 2020 on that graph. So we're really outside anything really that we've had for a long time. Down the bottom left, I've put that it's currently about 425 parts per million carbon dioxide in the atmosphere. And as many people on social media do, I put, when I was born, I put 314 ppm. I think I'm cheating. I think it was actually slightly less than that, but I didn't really want to know exactly how old I was. Also, to put this in the context of, if you like, thousands of years or hundreds of thousands of years, we're really outside anything that our species or even our genus has seen before. I'm not going to go into why we know it's fossil fuels. That's for someone else to observe on. So I'm going to talk about how plants respond to carbon dioxide. And they do this in 2 ways. One is on the direct effect of photosynthesis through the process, through fixation into sugars, and the other is the indirect effect through the effect of climate change. And there we've got 3 different scenarios of what the world will be like in terms of temperature change, rainfall, and so on. So I'm going to focus first on photosynthesis and basically the process, and I've kept this fairly straightforward because I don't know how many people know any biochemistry or anything about plants, but you have carbon dioxide and you have water, and you zap it with some sunlight. Actually, you don't zap the carbon dioxide and water with sunlight, but the plants absorb the sunlight, create electrical energy, which is translated into chemical energy that can then catalyze a series of reactions that results in the accumulation of starch. And it's all catalyzed by this enzyme. We call it ribisco for short, because ribulose 1,5-bisphosphate carboxylase oxygenase is a bit of a full mouthful. It is the most abundant protein on the planet and accounts for about 50% of the protein in the leaves of plants. So when you're looking at, when a cow is eating grass and getting some protein, it's getting it mostly from ribisco. So you think, okay, carbon dioxide's going up, starch will go up. More substrate, it'll go up. That kind of makes sense. And to an extent, that's true. If you just measure a plant and measure the photosynthesis, and instantaneously increase the carbon dioxide from, say, today's to 800, you'll get a higher photosynthetic rate. But what about plants that have spent their life growing at carbon dioxide? And you actually get a very different response then. And we do a lot of different experiments. You can grow plants in chambers, and the one at the left, that's not one of our chambers, but down the bottom left is one where we were growing taro and cassava in some chambers in Western Australia that we can set the CO2 and the temperature. There's open top chambers. So some people have been a bit critical of confined environments because the plants are in pots. You can grow them in glass houses. Then there's the gold standard, if you like, the free air carbon dioxide experiments. The one at the top right is what's called soy face, originally developed for growing soya beans in Illinois. And the bottom right is an Australian one called euc face, where they've had these huge chambers built around eucalypt trees. And the way these work is they have hose running around, a bit like garden hose, and it blows carbon dioxide off and uses huge amounts of carbon dioxide, but partly to cope with that and to reduce the amount. They have wind monitors. So if the wind's coming this way, you'll get more blowing on that side. And if the wind's blowing that way, and so on. So that it can keep it pretty even inside. And you can regulate the amount of carbon dioxide that the plants do. So that's how we know. So the predictions, and on the right here, we've got some photos. There's a colleague, John Evans, measuring the photosynthesis, what we call the instantaneous photosynthesis rate. If you want to look at something like cassava that's big, how big it's grown, you need very strong people to help you move them so you can actually weigh them. There's a lot of messy stuff, washing out roots. And then if you want to do chemical analysis, there's just endless samples. But the predictions for changes at elevated carbon dioxide is that higher carbon dioxide will increase photosynthesis. You know, the whole carbon dioxide fertilization, CO2 is good for plants thing. The plants should be bigger, they'll have more soluble carbohydrates, and also increase in some of the high carbon containing anti-nutritional factors like phenolics and tannins. And that's been shown to be true. But they also have a more efficient use of nitrogen. And the more efficient use of nitrogen means that actually they don't invest quite as much in rubisco as you'd think. And as a result, the leaves have less protein in them. And again, this is not universally true, but widely true. So here's some data that I put together years ago, and I keep looking at new papers, and it's all just still the same. So if you just get all the experiments that have been done on plants and look at this, is called the yield multiplier. So if it's above 1, these are combination, all kinds of different experiments. If it's above 1, the plants have had more yield, if you like. This is crop plants particularly, but you know, whatever, biomass, grain yield, all of that. And if it's below that, then they're smaller. So we can see everything's on the line or above. So these would be gains and these would be losses. But it's very variable. And for some plants like sorghum and actually maize, doesn't make much difference. So, other plants that are more woody, like cotton and trees, they have a bigger effect. And then sometimes if they're well watered or if they're droughted, you may or may not get different effects. And the carbon dioxide, so if you double the amount of carbon dioxide, you don't get twice the rate of photosynthesis. And the plants don't put on twice the biomass, and you don't get twice the amount of grain yield. It's all moderated and damped down. And the reason for this is that if we go back to this, is that this enzyme rubisco is promiscuous and it flirts with oxygen. It doesn't just flirt with oxygen, it has full-on affair with oxygen. And this has consequences. So for about every 5 or 6 carbon, this value has changed over the years, but this was a paper published about 3 years ago, so I figured they kind of knew. For about 5 or 6 molecules of carbon dioxide that go to make a sugar, about 1 or 2 molecules of oxygen there's various reasons. I'm going to say wasted. There's other things that could be said, but they get consumed as well. So rubisco is really inefficient. And the easiest way to make rubisco efficient is to add more carbon dioxide. So if you take a plant and you clamp one of those things on, like we have John in that picture, clamp one of these on and we can put the amount of carbon dioxide over that leaf higher than it is today. Plant's got lots of rubisco, photosynthesis rate's really high. But what happens if a plant is grown at high carbon dioxide? They actually invest less in rubisco because they can grow bigger with less rubisco. So they don't grow this much bigger, they grow this much bigger. Yep. No, it's not. So this is, you really get this in the C3 plants because rubisco is functioning at the more or less the concentrations of ambient air. With C4 plants, they have a carbon dioxide concentrating mechanism, which is already higher. And so you don't get that. You don't get the same effect. What's outside doesn't have the, has a small influence, but not a great influence. And so because you get this acclimation and growing, the plants don't grow, they grow better and they grow bigger, especially woody plants, but they don't, are not as big as you'd predict. In fact, and the plants make much, much less rubisco, which means they have much less protein in them. So just to explain here, this is the amount of protein in the grain or the part you eat. And again, this is old data, so I checked up 9021 paper. It's basically the same, so I didn't redraw the diagram. Again, this is oriented. Here's the change in protein of plants grown at elevated versus ambient CO2. If they had more protein, it would be on this side, so nothing has more. So on this side, they have less protein. Now, the reason for this is that the protein in the wheat grain or the rice grain or the barley grain or the soybean comes from the leaves. So as the leaves die off, and this is, everything's useful, right? I had my 3 years working on wheat in agriculture, and that's where I actually wrote a paper on how plants break down rubisco in the leaves to release protein for grain. It all comes back and be useful. So if the leaf has less protein, there's less protein available for the grain, and therefore grain has less protein in them. And this has already been observed. This is already happening as a result of rising carbon dioxide. It happens in potatoes too. I haven't looked into them too much. You don't get this effect in soybean, or not much. Does anyone know what sort of plant soybeans are? They're legumes, so they fix their own nitrogen. So there's no incentive, to use a teleological term, there's no particular advantage for soybean to make less rubisco because it's got plenty of nitrogen because it's a nitrogen fixing plant. But plants that aren't nitrogen fixing, nitrogen is always limited. That's, if you're looking at fertilizer in your garden, nitrate or urea or whatever is really what you need. And what's mostly it's been used to make rubisco. That's where most of it goes. So what's the result of low protein in wheat? I just snapped this photo at a talk I went to and then finally got the official photo. This work was done at the agricultural free air CO2 experiment in Horsham. And they grew this variety of wheat where there was no additional carbon dioxide, and the one on the right, which had an extra 200 parts per million. So this is at about 580 parts per million. At the moment, we're on track to get here by 2050. You can look at the bread, doesn't rise. To make a decent loaf of bread, you've got to have high protein. And also, as far as farmers are concerned, it's not just that, they get a much higher price for high quality grain. So they won't get as higher price for their wheat. Now, this is compensated to some extent because if you totally drought stress a plant or give a lot of heat stress, you'll get a lower yield, but the protein will go up. So there's a bit of interaction. So yes, you can either have low yields because you've droughted the plant from climate change, or high yields but low protein because of high carbon dioxide. It's not a good scenario to have these smaller, to have bread that can't rise. It also works in the way of animal feed. So if you are feeding grain to your chickens or your cows or whatever, you're going to have to give them more of that. And I was talking to someone who was a sheep farmer in Western Victoria one time, and I was saying, I was explaining about this, and I said, have you noticed any difference with your lambs? And he said, actually, our farm manager says that they actually now need to give the lambs have to be a certain size to sell to supermarkets. They actually need a bit of grain feed to get them up to that size now. We can't actually do it just on grass anymore. So that's completely anecdotal, never managed it. It's probably anecdotal on their behalf as well. But I think that this is an issue that a lot of people haven't considered. So my research is really on how plants balance and partition resources. So you've got a whole bunch of different resources, and just very simplistically, they can go towards photosynthesis, growth, or they can go towards defense. This could be toxins or other things, and that affects longevity. So that could be antifungal, antibacterial, could be anti-herbivore. So if you have resources that cut on photosynthesis, like low rainfall, high temperatures, for example, low light for other reasons, you would get less growth, and you think then those resources would be allocated to defence. There's a long-standing theory, and there's a lot of evidence for it. It's very simplistic. I think you call them heuristic theories. It just kind of explains stuff, really. And if there's things that promote photosynthesis, like high carbon dioxide or high growth, that might work. And if you have things that limit your defence, for example, some plants only make their defence compounds when there's danger, then you might get promotion to photosynthesis. Of course, the stuff that I've been doing, which I'm not going to talk about, is that this is just totally enmeshed with each other, and it's not as simple as this. But broadly speaking, it's still true. Now, I work on plants that are cyanogenic. They release toxic hydrogen cyanide when tissue is damaged. And you'd be familiar with these. Apple seeds, you know, that taste and smell of apple seeds, apricot kernels, they're not healthy. Just don't do it. Recently, a few years ago, a guy was admitted to hospital in Western Australia for taking a few apricot kernels every day because he thought it'd make him well, and he collapsed. There's about 30 types of eucalypt, cassava, which you were saying you grew up with, and bitter almonds particularly. So sweet almonds don't make it. It's just one gene different, bitter almonds do. And that smell of marzipan is actually the smell of cyanide, and you can smell that in your apple seeds. It's a normal apple with lots and lots of seeds in it. I'd probably, if I ate the core, I'd probably only need about 30 if I crunched it up really well to kill me. I also worked out that I use as my standard the sugar gum outside University House at Melbourne Uni, if you know that tree. So 30 leaves of those finely ground up and eaten all at once would be enough to kill me. So it's about 1 milligram per kilogram body weight.
These are 30 apples.
30 apples. Oh, cores, whole cores, yeah. But yeah, so the way it works is the plant would kill itself with cyanide. So it's not, and cyanide's volatile, so it's not going to be storing cyanide. Cyanide's very popular at the moment. About every murder drama at the moment seems to have a cyanide poisoning right now. And it is very toxic. But the plants store it as a cyanogenic glycoside, which is this compound here. So here's the cyanide group, which is a triple bond. There's a couple of R groups, which come from various amino acids or a hydroxyl group. And there's a sugar, and that's stable in the plant. If it wasn't stable, the plant would kill itself. But separately, and located separately, is that it has an enzyme which can cleave off this sugar. This becomes unstable, goes through a couple of steps, and eventually releases hydrogen cyanide and either an aldehyde or a ketone, depending on what these R groups are. It also releases sugar. So, and some parts, for example, in Nigeria, they sprinkle a bit of crushed up cassava over their food to make it taste sweeter. Cyanide binds to cytochrome oxidase in the mitochondria, which causes death to aerobic organisms. So the first experiment I did for my PhD, and which was published in '98, so I didn't complete my PhD till '99. Was we grew sugar gum seedlings in the greenhouse on the roof of the botany school, and at either 380 or 780 ppm. 380 was ambient in those days. What a joke. It's 425 now. And we measured biomass, photosynthesis, and so on. Now, just looking at those plants, you think, wow, the ones at high CO2 are taller. Yes, they were. They had more biomass. Something else you might notice, which I had never thought about beforehand, they've got less leaves. they actually don't produce as much leaves. They put the biomass into making deeper roots and not as many leaves because they can just get enough carbon dioxide without that many leaves. And the leaves tend to be thicker as well, so they're harder to digest. So we measured a whole bunch of stuff, and this sort of summarizes, the work. If the size of the pie is the amount of nitrogen in the plant, so nitrogen equivalent to protein, so you can estimate protein's about 6 times the nitrogen in terms of mass. And this is divided up by various proteins. These are in the leaves, rubisco. This is the hydrogen cyanide, and this would be various allocated to nucleic acids and some other things. When we grew them at elevated carbon dioxide, the whole pie is smaller. And interestingly, the rubisco, total rubisco, like milligrams per gram leaf, was 40% less. So it's a lot less. And as a proportion of the total nitrogen, it was even smaller. And the proportion in the total amount of cyanide was higher. And I'm using cyanide as a shorthand for hydrogen cyanide or cyanogenic glucosides. The cyanide was increased. And so you can see that this is clearly not healthy. And for the plants. Now, one of the things, so we got out and we talked about it. They also had more phenolics and tannins, which are anti-nutritional factors, say for arboreal mammals like possums. You may not be sorry about that, but koalas, for example. So my conclusion of this is carbon dioxide may be good for plants, but it's not for the animals that eat them. And what's hidden by the image here, my picture here, is why don't people listen? Because we talked a lot about that. So here's me during my PhD, aged like 40 something, doing a video for the ABC. You can still online. Actually, I put it online, so it's there. I got permission. Cut it down. We had some nice things, how it works, how the whole photosynthesis works. We had articles in the Australian, various other newspapers, and I was thinking, we said, even with that, like, let's just be fair, koalas do not eat sugar gums. They've got cyanide in them, and they know that they would kill them. So they don't, but other insects do. But just on the basis of the phenolic changes, we worked out that by 2050, there would not be enough protein in these leaves to support koalas. Now, this is not, so you have to give them extra food. So the protein in the leaves, like to support insects, you have to have more than 1% leaf nitrogen. To support koalas, you need to have more than 2% leaf nitrogen. If you grow the plants in, a typically poor soil and at high carbon dioxide, the leaves will fall below 2% nitrogen and therefore would not, extrapolate that, wouldn't be able to support arboreal mammals. I mean, eucalypts are crummy food anyway. So just to move on now, just want to just talk a little bit about some crop plants. What's the time? Okay, we're going well. I said something about the sheep. I said something about the wheat. Now I'm going to talk about the plants that I have worked mostly on, which are sorghum and cassava. And as you pointed out, some plants have a different photosynthesis system, like which sorghum does, which makes it less sensitive to atmospheric carbon dioxide. So I'm going to talk about both of these because they contain hydrogen cyanide. And when I left Melbourne Uni and went to Monash, I thought, well, I can't work on eucalypts anymore because my old group was still working on them, but I wanted to work on these cyanogenic compounds. And I was very aware that this became was a social issue. I think it's quite interesting because when I was growing up, my parents had this very much a world view of doing good. And, I think they'd even thought themselves that they might go and work in, a developed country or maybe in parts of Australia and work in public health. So I've kind of grown up with this sort of idea, and it was a natural transition then for me to work on crops, especially after my few years of working in agriculture. So first of all, I was working on sorghum, and this started about because a guy from a company called Pacific Seeds got in touch and said, could I test their varieties of sorghum for hydrogen cyanide? Because it's a problem in Queensland. It's a major grazing crop, forage sorghum. The grain is okay, so don't worry about that. but the leaves can have high levels of hydrogen cyanide. And it's very hard for the farmers to predict. So they might look out, it's hot and dry. The only green thing they can see is sorghum, and they don't know whether it's going to kill their cattle or not. Or if it doesn't kill them, maybe they'll get a disease called staggers, which is where they get a paralysis in the limbs. So they want to know, and the testing that goes on takes too long. They send it away to the labs and get it back. And, it's often several days later, and it can actually fluctuate. If it rains or you have a hot day, it can vary a lot. So he got in touch with me and, basically, when can you work out whether a cow can eat it? And we've done a whole series of experiments on this, and I'm not focusing on that in today's talk. But basically, if water, the resource, is limited, growth is limited. And so you get an increase in these defense, in this case, in the hydrogen cyanide or the cyanogenic glucoside in sorghum. Now, you'd think that could be just because the plant's smaller, right? So it's concentrating down, and there is an element of that. So the plant's smaller, it's making the same amount, so the concentration of the toxins are higher. That's true. But what we've actually shown is that the plants all the genes in the pathway, biosynthesis pathway, are all upregulated. And we can actually induce that by using other things to pretend it's a drought, like spraying them with various plant hormones to make them think, that stress hormones like abscisic acid and methyl jasmonate, and you have the same response. They'll increase this. So it's not just a dilution effect or a concentration effect. So to summarise in this next little bit, like 15 years work, it turns out there's a strong effect of water. There's also effect of temperature. And there's little carbon dioxide effect because sorghum has this carbon dioxide concentrating mechanism that's already more efficient. In fact, at high carbon dioxide, they actually use more efficiently. And if anything, become slightly less toxic if you only had that higher carbon dioxide. But we were then working with Peter Stewart, who was then at Pacific Seeds and then became Advanta. We developed a completely non-toxic version, and we used non-GMO methods. We used mutagenesis. Two reasons why we did that. One is at the time you couldn't transform sorghum for love nor money. It was very, very hard to do. Even now it's still pretty hard, but you can do it. Many people do it routinely. And the other is that dairy farmers, who are a big consumer of forage sorghum in Queensland and Australia, would then and probably still be very resistant to using a GMO product to feed their cattle. So we used a mutagenesis system. It takes a long time. You screen a lot of plants, 50,000 plants, and you find the pathway mutations in the pathway you want. Out of 50,000 seeds treated, we found one line with a mutation in the right gene that knocked it out. So all I can say is, thank you to the Australian Research Council for funding such a risky project, and that it actually came off. And it's now in commercial production, which is great. And this is in collaboration with other colleagues at Copenhagen. Now, it's really difficult to predict prussic acid or cyanide levels where there's many variables, and that's why knocking it out was an option. So young plants, like young sorghum plants, are very toxic. Young plants in general are much more toxic than old plants. Probably they're more vulnerable to predation at that time, and so there's been an adaptation to it. There's a few exceptions, but nearly always that's the case. So farmers know you don't graze cattle on a crop that's less than half a metre. You just don't do it. We know that drought increases. This compound here is Durin, which is the cyanogenic glucoside in sorghum. And we know that if we add extra nitrogen to the crop, it goes up. So you can give farmers a bit of a plan and say, okay, if you've recently fertilised, the plants are young and there's a drought, don't do it. How does carbon dioxide, but drought also affects nitrogen. So if you drought them, plants actually have more nitrogen. They actually concentrate it up. Carbon dioxide not only interacts with drought, interacts with nitrogen, and in this case has a small direct effect. So it's actually very difficult to predict. And you think, wouldn't it be nice just to test and use some little test papers? And we've tried to get this going for sorghum. We've got them for some other species, but we haven't got the one for sorghum working. If someone wants to do it, I'll tell them I know exactly how it should be done. I just can't do it. So we knocked this out using a mutagenesis method. And as I said, that's now in production. Okay, so let's switch now to cassava. Cassava's a staple food for 550 million people, probably more. It's 40% of sub-Saharan Africa. It's a staple. There's 1 billion people in the world eat it every day. And it's very cyanogenic. Every part, the leaves, the stems, the roots, everything. And people ask me what it looks like, and I say it basically looks like a marijuana plant with sweet potatoes at the bottom. That's kind of what it looks like. And so this farmer in Mozambique is very proud. He thinks this is a big harvest. It's not huge, but it's not bad. And it grows in very poor soils and difficult conditions. And these are the yields in 2021, and you can see high production in India, through Indonesia, Africa, and so on. So there's a lot of, this is tonnes per hectare. So we can certainly improve on the production, and the FAO, Federation of Agricultural Organisations, is that what it's called in the UN? Is that what it's called? I've got the right name? Yeah, Food and Agricultural Organisation. They are very much into increasing production of cassava and having more and more cassava. However, there needs to be a caveat on that says that is great. It's a fantastic crop. It's now grown very widely in the Pacific. It was actually only introduced there about 60 to 70 years ago. People who go to the Pacific would find that hard to believe because you see it everywhere. But it turns out that the knowledge of it, the potential of it causing problems from cyanide, is moving slower than the knowledge than where it's grown. So it's the spread of cassava growth is increasing faster than any knowledge about the problems with it. So what happens if you eat, and this is particularly a monotonous cassava diet that's high in cyanide. So these children have a disease called konzo. These children are actually all from the same family, and there's some really interesting research going on with this. The one on the left would be mild, he can walk. The one in the middle is moderate, he's using a stick. The severe will just get around on his backside. And it can come on quite quickly. So if they eat a lot of cassava, whether it's high in cyanide, this can come on just in a matter of a few days or a week, and it's permanent. So that's not a paralysis. I think they call it I can't remember the word. Paraparesis. That's right. I think that's the technical one. So you can get acute cyanide poisoning, as we know from Agatha Christie. That depends on the dose, how much you're eating, and your other parts of your diet. So I could eat 30 leaves of the sugar gum growing outside University House at Melbourne Uni if I ate it very slowly over 24 hours. But if I ate it all at once, I couldn't. It can cause vomiting and dizziness, does so regularly, and of course it can cause death. Chronic conditions include things like goiter. I can come to that in a minute, why that might be, and also increasing evidence of cognitive impairment. So a certain amount of cyanide in the diet is going to reduce, presumably the hypothesis is reducing oxygen delivery to the brain, and therefore brain development, again, particularly in children. And then we have konzo. There's epidemics of konzo. There's an epidemic, well, there was 6 months ago, an epidemic in Mozambique where it's been very dry. There's environmental factors, tends to be associated with droughts. There's social factors, and some individuals are more affected than others. And there's some really interesting research that looks at possibly one reason why different people in the same family may get to different degrees, maybe because their microbiome may be different, or there may be some genetic differences in them. But it's very, very new research. So humans can detoxify cyanide, and it depends on the rate and dose. Plants actually make this enzyme too, rhodinase. Obviously in plants, it's not taking place in their kidneys, but most plants will have rhodinase, and cyanogenic plants have more than non-cyanogenic plants. It catalyzes a reaction between cyanide and thiosulfate, which is derived from sulfur-containing amino acids, particularly, methionine and cysteine. I've spelt that incorrectly, sorry. That produces sulfate and thiocyanate. And thiocyanate is much less toxic, and it's excreted in the urine. And a system that's been used very widely for testing how much cyanide kids in villages in Africa are eating is by testing the amount of thiocyanate in the urine. Interesting, just recently there's been a big outbreak in Democratic Republic of Congo. I was speaking to a guy there who was doing some testing, and they'd ordered the wrong kits for testing thiocyanate, and they were actually testing for cyanide, and they were measuring, and there was just an outbreak just happening. They were watching it happen in the week they were there. They were measuring free cyanide in the urine of these kids, which was incredible. So this was first really put together by a Melbourne doctor called Julie Cliff. And she moved to Mozambique in 1976. She was public health. She studied Melbourne Uni, did medicine there. And she became aware, well, was notified because she was working in epidemiology of this outbreak of a disease. And they didn't know what it was. And there was a man called Hans Rosling there who you might have heard of, who became quite famous later on for his data analysis. Unfortunately passed away far too young at 68. And Hans got in touch with her. He was way out in the field and he said, Julie, you've got to come and have a look at this. There's something happening. And they went around, they kept finding more and more cases, more and more cases, more and more cases. And they couldn't work out what it was. They were doctors, they thought it must have been a disease. Was it polio? Was it they couldn't work it out. And the people kept saying to them, it's something to do with the food. It's something to do with the food. And then they realised they got some samples and they managed to get them tested for hydrogen cyanide of the food, that is, and the urine, and they found that, in fact, it was high levels of cyanide in the diet.
And that was in 1981.
That was in 1981. So when they looked back over the historical records, there are actually records of a similar outbreak. And in fact, the name konzo was coined back in the 1950s. So they did the right thing and went all the way back to, and there's been records of it, and probably earlier as well. So why was there an outbreak? She wrote up several papers on this. First of all, there was a drought. So everyone was relying on cassava. And secondly, there was a war going on, right? So a big civil war going on in Mozambique at the time. So people, were limited in what they could do with their food supply. And the third thing is that it was only, those of you who are younger won't remember, but people my age will remember the Portuguese had their own little revolution big revolution in 1975, and they left their colonies, which was, going to happen anyway, but they did. But one of the things the Portuguese had in place is that they had a ban on what was called growing bitter cassava. They're the ones that have very high levels. And that constraint had fallen away with the removal of with any kind of governance, really. For several years, there wasn't much governance going on at all. And so people were planting more bitter varieties as well. So there was a number of different reasons. Now, later on, a few years later, a Howard Bradbury, who's pictured here, he passed away recently, about 7 years ago. He did work on the cassava flour in the marketplaces in Maputo with others there, other Mozambican colleagues. The World Health Organization recommendation for cyanide in food is 10 parts per million. In a normal year, if you go to the Mozambique markets, it's about 15 to 20 parts per million, and we were there in a normal year, and that is about what we measured. In a drought year, it's over 200. So clearly the plants have got more in them. Now, I don't know how it was connected. I think Howard reviewed one of my papers. But anyway, he gave me a call and said, you need to get in touch with Julie Cliff, because I think what you've been finding with cassava is interesting. Because what we've been finding, so first of all, I'll come back to this. What we've been finding is that we've grown some plants, cassava plants that were just because we thought it would be interesting because it had cyanide. We didn't really know much other than what we'd read about what was going on. And we had plants that were watered, plants that were droughted. Here's two honours students who did the work, who looked at different parts. One looked at the genetics, one looked at plant growth. And there's the growth of the plants, and we had plants where they were only young. Some of them were well watered and some were droughted. How do you know? You weigh the pots every second day and make up the water. It's a huge amount of work. Half the plants were re-watered after 2 weeks, and then we grew them for another 4 weeks. So some were droughted for 4 weeks, some for 2 weeks. and then re-watered, and some watered all the time. And we found that in the tubers, just for reference, here's the WHO recommended level. This is just growing in our glasshouse at Monash. We had 1,200 parts per million in the droughted plants, about 200 or so in the watered plants. But we actually found that if you re-watered the plants for a couple of weeks, actually the level dropped right down again. So that's something that really hasn't been looked into again, but I think it's something that could be considered as a control mechanism. Anyway, Howard was aware of what we were doing, and he got me in touch with Julie, and we went and visited Mozambique and did a series of field studies over there as well. Now, the reason I've come back to this one, of course, you can process cassava to make it perfectly safe. If you're not processing it, you're probably eating lots of fish and other things with it, which will give you enough sulfur to detoxify it. Of course, the sulfur you're using to detoxify it is sulfur you don't have to make thyroxine. That's why you get goiter. But you pound it up, pounding it together. So you can see the woman on the left is pounding away, and she'll make sort of chapatis with that. That will get rid of it. You can soak it. Some of them in DR's Democratic Republic of Congo, they mostly just put it in a little bag and chuck it in the river for 3 days. But of course, there's a war on. So what are they doing at the moment? 1 day if you're lucky, right? So they've reduced that length of time. You can just dry it. That'll get rid of a little bit, particularly if it's shredded. One of the problems, grating is fantastic. But this work is done by women and often by young girls. And as they do it, they get dizzy because the cyanide's been released as they're grating it, and they become quite dizzy. So that's a problem that needs to be addressed. And I think with increasing commercialisation, as you see in Nigeria, where that stuff's done in factories rather than by individuals, I think that problem could be avoided. And then in South America, the plant is originally from South America. We think of it as African, but actually it is a South American plant. They've been growing it there for at least 3,000 years. The Amazonians have this fabulous system, a little thing called a TPG. I've got a little model one here. I'll just do one for the people who are watching online. But basically, they're about this big, right? About 1 metre long. But you stuff it full of your pulp. Then you hang that on a branch, and you put another branch in the bottom, use the lever. So you fill it up, it's all sort of moist, and you just press it down, all the water runs out the outside, and it removes about 95+% of the cyanide. Absolutely brilliant system. Not used in Africa, possibly because of the large amount of water that's required. So why doesn't everybody process all the time? Well, it's a time commitment. It's not always needed, so not all the plants, not always. There's varieties that don't make as much. Might be social unrest. The flavour is different, and some people like that bitter flavour. And there's also the knowledge. And Howard went on in his retirement to develop the wetting method. And this is a system that works very, very well. He also developed this system of test papers, which are available for free for people in developing countries or anywhere else. You have to pay for them, where you can pick rate papers, where you can, works a bit like litmus test, and you put it in, and it has little discs in it that has little tubes with the enzyme, and it breaks down, and in about 4 hours you get your result, and you have a standard in it. It works incredibly well. So he invented that. It's all published, public domain. You can make your own if you want to handle picric acid. It's easy. Picric acid's explosive, but there's some of our samples in Mozambique. So here's the method. They've been translated into about 50 different languages, and it says basically you grind up your flour, you put it in the bowl before you cook it. This is obviously not where you're roasting whole tubers, but you're cooking flour, and then you spread it out and leave it for about 4 hours and then cook it, and 97% of the cyanide or even more is reduced. So this is highly effective. Has to be done when the temperature is over 25 because cyanide doesn't evolve and is actually not volatile at lower temperatures. So I can't use it in the highlands of DRC and Rwanda, for example, because it's too cold. And he also started this thing called the Cyanide Diseases Network, which unfortunately was difficult to support after he passed away. And part of that was so that African scientists and people from developing or first world countries and lower income countries could publish their work. So we use these test papers and other methods to work out about some Australian products, and we've just repeated this. So tapioca, don't worry, if you're buying grated cassava, not a problem. What are some of the other things? Well, if you buy whole frozen cassava, the flesh, it's not bad. The peel is incredibly toxic. The peel of a fresh plant would be much, much higher than that again. How many Australians know they have to peel their cassava? Like zip, nobody knows, right? But really, I probably shouldn't say this, will be the subject of the next TV drama. We also found imported chips varied between about 25, which is safe, to over 200 parts per million. We worked out this particular brand had enough cyanide in it that if a 2-year-old child ate a whole packet, they'd die. So Australia is one of 2 countries in the world that have introduced legislation that food is supposed to be less than 10 parts per million. This came in 2011, 2010. Between 2005 and 2010, when we measured veggie chips, for example, they were all about 70 to 80. That's not massively high. One of the packets I brought home from like an Air New Zealand flight from New Zealand was about 85. It's not here because I didn't replicate it because I ate the other packet. But in 2011 and every test we've done since then, veggie chips are below 10. So they're doing the right thing. How do we know people do the right thing? Howard and I used to go out and test. Howard's passed away. I'm retired. There isn't actually any monitoring. So I think it would be great if we had some monitoring for this. But as I said, we just repeated this with a whole lot of cassava products. And generally, it's not bad, but you do have to be a bit careful for some of the imported ones, particularly if it's chips that are a whole piece that's been cut. So if it's been mashed up and made into a chip, which mostly is veggie chips, some of the brands have that, they will be much safer than ones that are just a whole piece because they may not have had enough exposed surface. So we also then went on and did a series of free air CO2 experiments. And there's just one I'm going to show you now. This led on to a whole thing called Cassava Face. So it's called FACE for free air CO2 experiments. And it was set up at Soy Face. And together with my colleague Tim Cavagnaro, who's at Flinders now, we grew cassava in Illinois. If you've been to Illinois, it's quite cold in Illinois, but we worked out with the people who worked there from climate models that if we could get the seedlings in May, we could harvest them in October and they'd have a good 6 months of good growth. So you can see by July, these plants were huge. And initially, they were just little clearings in their long-term maize experiments. So there's all the maize around. It sort of looked like from the air, I'm sure it just looked like little clearings of dope growing in the middle of a field or something, but it was cassava. And his one of the postdoc there working on it, actually doing the photosynthesis measurements was that tall. He had to grow it up to like that. And here you can see the leaf sampling that we've done. Now, this has been repeated and there's a whole set. They've now got a thing called Cassava Face, which is the same thing, but they just grow lots and lots of cassava. And grown lots of varieties and added in some other treatments. But I'm just going to show you the results of this first experiment, which are a bit different from what we found in some of our very early studies. So at the time, the ambient CO2 was 390 parts per million, and they were enriching to 580 parts per million. They've since upped the today's level. Today's level is obviously much higher. It's 425, and they've upped the enhanced one to 650 because there's not enough difference now between ambient and elevated, which is a bit sad. But you can see, first of all, you look way, way more tubers. So it turns out cassava doesn't do this thing of getting less photosynthesis as much. Why not? Because it has tubers. All that carbohydrate that's been made in the leaves is being transported to the tubers. So the leaves never get this build-up of carbohydrate, of sugars and starch in the leaves. That's the signal that signal to the plant, don't need to do so much photosynthesis. Scale back your rubisco. That's from the accumulation of carbohydrates in the leaves. But plants with tubers don't do that, don't have that issue because so much of the starch is pulled away. It does it a bit, but not as much. So you do get a big enhancement in yield. But what we did find, both the tubers and the leaves had less protein in them. So they did have some acclimation, if we call it, to CO2. So they did have some downgrade, but not as much as some others. And they had the same amount of cyanide. So if you're looking at cyanide to protein ratio, they're definitely more toxic. But as some of us were discussing before, the protein levels in cassava are so low that you really need to be getting your protein from another source. So this is why if you have a monotonous cassava diet and you're not getting protein from another source, it can be so toxic because the plant itself makes very little protein, and under elevated CO2, that would be much worse. So what's really needed is education, I think. So these theories of resource allocation have helped us predict in many ways that when we've got resources that are limited, we limit growth and we have more defense. At elevated CO2, we enhance photosynthesis, and you think we get less defense, but in fact they acclimate. So they don't increase that photosynthesis much, and some goes down the other side of the pathway. So why not just knock out the capacity to make cyanogenic glucosides? Makes sense, right? So this is a project done by Jess Lyons that I advised with from UC Berkeley. This was January 2020, I just like to say. We snuck in the trip. This is the pathway for synthesis in cassava. It actually has, I didn't show you the other pathway, but sorghum has one gene here, so it was relatively easy to find mutants. But she's had to knock out both those genes to get rid of the cyanogenic glucosides. There's two different ones in cassava. So she's knocked that out, and so she found that if you knock out one gene, you get a reduction. If you knock out if you knock out the other gene, you get a bigger reduction. If you knock out both the genes, you get zero. So you can make a zero cassava. She didn't see any effects on growth. So early studies had shown if you knock out the cyanide completely, the plants don't grow so well. And that's been my experience. The zero sorghums and zero everything else we've grown just have lost a bit of the edge when they're young. But she has found no difference. So that is really promising. And she was set up to do field trials in Nigeria, but with COVID, they didn't go ahead. I don't think they've been done. So why don't people just plant sweet, that is, less than 100 ppm cassava, rather than bitter, which is more toxic? Why not? That makes a lot of sense, like the Portuguese used to recommend. In fact, most of the varieties in the Pacific are sweet varieties, not all. Part of it is the lack of knowledge. Some of it's the availability. But there is a preference for growing bitter for cassava. And I thought this was kind of a bit of a myth. I thought maybe they're more resistant to pests and diseases. I've got about 3 more slides, by the way, if you're wondering about time. But there's been studies showing that they're less likely to be taken by animals, particularly baboons, work in Malawi. It also, in Malawi, they showed that it gave women control over the food supply. So the men just wouldn't come in and just dig up your cassava and take it off, or the soldiers or whatever, because they had to be processed, and that's women's work. So the women have control over it. So that's another reason. But there was also this thing that bitter cassava yields better. And we've been trying to get on top of this, but our early experiments are showing that confirm that bitter cassava may be more tolerant of drought and may be better at recovering. So what I've called, in a very, everyone's into like colonial science, how bad it is, talk about that. I'm saying, it's just a myth. Actually, there is many reasons for growing more toxic varieties. So I don't think growing a knockout cassava is going to work because people won't want to grow it. Now, humans seem to have a fatal attraction for cyanide. Over 30 species of crop species make cyanide, and it's only about 5% if you just have different wild plants. Sorghum leaves, domesticated sorghum, the leaves have 1,000 times are more cyanogenic than the wild relatives from Australia, mostly Australian. And we've also got work we did with the Berkeley group that shows that cassava probably became more cyanogenic with domestication even 1,000, 2,000 years ago. These are many of the people who have actually do the work. So how can we make cassava safe now? First of all, don't eat it. That's ridiculous. Of course, people are going to eat it, but we need to improve diet. Lifting people out of poverty would be the #1 thing. Easy. billions of people. We lift them out of poverty. Using GMOs or knockout cassavas, problems with GMO acceptance, not widespread, and also there could be some trade-offs with performance. And the answer is really processing. So we've got to get the message out there. So what I've done, I've been wanting to do this for years, and now I've retired, I've actually got time to do it. I've set up the International Working Group for Safe Cassava. We tried multiple times and we nearly got it off the ground in 2019, and then we had the COVID restrictions, but we finally set it up in 2023. Jan Elliott's the woman at ANU who makes all those kits for free, I might say. She's a volunteer. So all those kits that everyone around the world uses for testing, everyone uses in their experiments, are all made by Jan for free. I mean, she's free, her labour's free. Julie Cliff, who comes back to Melbourne regularly, and me, and everyone's unpaid except for Kira, my research assistant, who I have enough money to pay for half a day a week to help me organise it. So it's not great, but we're getting there. And what we've done, we've actually now got all the names from Howard's old cyanide diseases network and created this Safe Cassava network, because we sort of had to change the name. We've even got a logo. And a domain name. And the aim is to foster collaboration between those working on cassava and konzo. We have webinars. They're on the Global Plant Council YouTube page. There's some advantages in being the past president, and it's supported by the Konzo Prevention Fund via ANU. And we're actually having a symposium, our first symposium, in about 2 weeks' time. It's free. Anyone can log on. You probably can't get the QR codes from there, but if you go to the ANU website for konzo prevention, you can find it there. We've got Torquil Tessela, who was Hans Rosling's PhD student, worked on konzo his whole life, looking at the neurology side with a whole series of speakers. They're just chairing the sessions. We're looking at cassava the plant. Carlos was a major plant breeder of cassava back in the days. We've got a group from Mozambique talking about public health rehabilitation and how that's working in Mozambique. And Louise Abiyamo, who's at the University of Greenwich, but spends half of every year in Nigeria. She is Nigerian originally, and she's talking about food systems and has a panel that we're going to some of that. Now the times are all worked. It's just a couple of hours each day, because obviously no one's not going to sit on the whole thing. It'll be recorded and they'll be available afterwards. So please log on if you want, or you can watch them afterwards. So I guess I kind of come full circle. I grew up thinking I was going to be a doctor, because that's what my family did. I grew up thinking I was going to do good in the world, because that's what my parents' view of the world. And a few years ago, I went, I was back in Africa. I mean, I've been to Africa many times now. So I think this is the tale of the story is, sometimes things don't work out what way you think. You think you've got a goal and you've got these milestones and everything like that. But as my kids are fond to tell me, they gave me this picture when I was a bit stressed. Life's an adventure, not a worry. So with that, thank you for your attention.